Namaskar, welcome to PK's classes. Today we are going to study polymerase chain reaction. We also know it as PCR. This PCR is a laboratory technique, an in vitro technique, a cell-free technique. We do not need a cell. Just we need a sample of DNA, even one molecule of the DNA, and we can amplify the DNA through this technique. So it is a DNA amplification technique. So by this technique, we can synthesize multiple identical copies of any DNA of interest. So if we define this PCR, so PCR is a laboratory technique or in vitro technique or a cell-free amplification technique to generate multiple identical copies of DNA of interest. So what is its principle? As you know, from one DNA we can produce two DNA. From one DNA we can produce two DNA, and first there will be unwinding of the DNA double strand into two single strands. Then each strand will act as a template and two DNA are produced. So in PCR the same principle is applied using temperature. So if we use temperature 95 degrees Celsius, then there will be denaturation of the double stranded DNA into two single strands. So 95 degree Celsius. Then if you lower the temperature to 55 degrees Celsius in presence of the primers, then those primers will be attached to the 3' end of the each strands. We call this step as the renaturation or annealing. Then if we provide the enzyme DNA polymerase, which can withstand the temperature up to 95 degrees Celsius, so thermostable DNA polymerase if we can provide, then each of these two strands will elongate in presence of the deoxyribonucleotides, the four ones, deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytosine triphosphate, and deoxythymidine triphosphate. So if we provide all these four deoxyribonucleotides, then the two strands will be formed. So two DNA strands we can get. So after one cycle we will get two. After two cycles we will get 2 into 2, 4. So one cycle consists of denaturation, where the temperature is kept at 95 degrees Celsius. Then we lower the temperature to 55 degrees Celsius and there will be annealing, the attachment of the primers. The process is called renaturation. Then we increase the temperature to 75 degrees Celsius and there will be synthesis of the DNA in presence of the DNA polymerase and the deoxyribonucleotides. So since this process occurs, or this cycle of events of consisting of denaturation, renaturation and synthesis occurs due to variations in the temperature, by increasing the temperature, then lowering the temperature, then increasing the temperature, we call it as thermocycler. So the PCR instrument is also known as thermocycler. Thermo for temperature, cycler, one after another the cycle is repeated. That's why we call this as a chain reaction, polymerase chain reaction. So a chain reaction of or a cycle of events occur, and Each cycle consists of 3 processes: denaturation, renaturation, and synthesis, and they are repeated one after another. So after one cycle we get 2 DNAs, then after 2 cycles we get 4 or 2 to the power 2, after 3 cycles 2 to the power 3, after 4 cycles 2 to the power 4. So after n cycles we will get 2 to the power n number of DNA. So just imagine, say after 30 cycles, what will happen. And each cycle takes about 3 to 5 minutes. So accordingly, if we put all these ingredients in the PCR, then say and keep it for say 1 hour or 1.5 hours, so 20 to 30 cycles will be there, and you can imagine how much number of DNA we can get. So you will get billions of DNA in a few hours. So what are the requirements? We just need the target DNA. The sample should contain at least 1 molecule of the DNA, and that DNA should have 100 to 35,000 base pair in length, and the 2 primers which are so we need a target DNA which is 100 to 35,000 base pair in length, and 2 primers. The primers are basically the synthetic oligonucleotides of 70 to 30 nucleotides length, and they should be complementary to regions flanking the target DNA. So flanking the target DNA means nearby to the target DNA. So if these primers are complementary, then they can start the process of DNA synthesis. So for that we need one DNA polymerase enzyme which is thermostable, which can withstand at a temperature up to 95 degrees Celsius, and the thermophilic bacteria like Thermus aquaticus provides one DNA polymerase. We call it as stack DNA polymerase, and Thermococcus littoralis is another source, vent DNA polymerase. These are the commonly used thermostable DNA polymerase in PCR. Then 4 deoxyribonucleotides like deoxyadenosine triphosphate, deoxycytosine triphosphate, deoxyguanosine triphosphate, deoxythymidine triphosphate, these are also required. So what will happen? First, let us means in the when we put all these ingredients in the PCR machine, the target DNA, we want to multiply this target DNA. So first the primers, first there will be denaturation when the temperature is raised to 95 degrees Celsius. Then we will provide the DNA will be divided into separated into 2 individual strands, 2 single strands by the process of denaturation. Then the primers will be attached. The primers will be attached to the 3' end, and so that the initiation of DNA synthesis occurs at the 3' end. So the primers will be attached, and the process is called renaturation or annealing when the temperature is lower to 55 degrees Celsius. Then the temperature is raised to 75 degrees Celsius in presence of the enzyme TAC DNA polymerase, and there will be synthesis of the DNA. So from one DNA we will get 2 DNA, and in the next again we increase the temperature to 95 degrees Celsius. The second cycle starts, and accordingly the cycles will continue until we stop it. So the reaction can be stopped, and the number of DNA will depend on the number of cycles. So the applications of PCR, number one is the clinical diagnosis. We can use PCR for amplification of the DNA, and then we can use that for diagnosis of viral infections, bacterial infections, cancer, and inherited diseases. So prenatal diagnosis can be done. Sex determination of embryo in humans and livestock also can be done. We can also use PCR for gene sequencing or DNA sequencing, gene expression studies, gene manipulation and gene expression studies, comparative study of genomes and classification of organisms, study of evolutionary biology or phylogenetics. We can use PCR in drug discovery, we can use it in study of antimicrobial resistance, and the most common use is in forensic medicine for identification of criminals. So if we have a single molecule of DNA from the blood stain or semen or hair, then we can amplify it and we can use it. So what are the advantages of PCR? Advantages like high sensitivity, very small amount can be used for detection. So highly sensitive, quick and cost effective, and can detect to the microorganisms which are less common, and we can detect like viruses which are less commonly known, that can be also detected through PCR. So disadvantages, they are having lower specificity as compared to cell culture, and though it is cost effective, but the supply cost, the machinery fees, even training is also needed, so training expenses are there. Then variations of PCR. So there are a number of variations, and one is the reverse transcription PCR, and the other is real-time PCR. These two we'll discuss. So reverse transcription PCR is required. PCR we can amplify DNA. So if we have a sample of RNA, then that RNA has to be converted into a complementary DNA by use of the enzyme reverse transcriptase. So this is added in addition to the PCR technique. So once the complementary DNA is available, then it can serve as a template for the PCR. So then the PCR process will be there. So we call it as RT-PCR, reverse transcription PCR, and you know the coronavirus is an RNA virus. So during this COVID-19 pandemic, RT-PCR was very common. You must have heard about this. So the RNA sample is first converted to its complementary DNA by use of reverse transcriptase, then the same process as PCR. So another variation is the real-time PCR, and this is a quantitative technique. So it is used for quantification of PCR products in different cycles. So for that we use a fluorescent compound like ethidium bromide, and the DNA binds to the ethidium bromide and emits fluorescence, and we can detect that fluorescence, and from that fluorescence intensity we can quantify DNA using a standard curve. So this is real-time PCR. Thank you for watching this video.